All right, I'm going to pick up again with the more details on the phases of meiosis. We're going to start with meiosis 1. Prophase 1 of meiosis 1 is going to be where spindle forms and the nuclear envelope fragments and the nucleolus disappears. That's all the same as mitosis. However, a difference is that the homologous chromosomes will synapse. What that means is they're going to form that tetrad that I was talking about in the last video, where they hold hands. So you have homologous pairs. They're going to come together. It doesn't show it here, but this one and this one will come together and hold hands in the center. And this one and this one will come together and hold hands in the center and form that tetrad. And remember, it's not written up here, but I'll add it. That's where crossing over occurs during prophase 1. Now in metaphase 1, that's where they're going to line up down the middle. I don't know why I can't write on here. Okay, so that's where it's going to line up down the middle in metaphase 1. For some reason I can't write it. So it should say on here that that's the law of independent assortment. Metaphase 1 right here, that's where it could be the chromosome from the mom on the right, or it could be the chromosome from the dad on the right. And the way that they line up on which one goes on the right and which one goes on the left is independent of any of the others. So it could be swapped either way for all 23 pairs. That's a lot of different combinations. And that's the law of independent assortment that we talked about last time. That happens in metaphase 1. So the main parts that you need to remember are in red here. Prophase 1, we had homologous chromosomes synapse, forming those tetrads, and crossing over occurs. Metaphase 1, we have homologous chromosomes line up in the center right here, and that's the law of independent assortment. Anaphase 1, we have chromosomes that are going to be reduced to haploid, and that's because, remember, Ana is the one that pulls them apart. So anaphase 1, they're going to pull them away. So the centromeres do not dissolve here. They're still stuck together. They're still sister chromatids held together by centromeres, but the homologous pairs are what are separating. So we went from being diploid up here to now they're going to be haploid. So haploid, that's huge. Chromosomes are reduced to haploid. That happens in anaphase 1. So all the big events that we've been talking about happen in meiosis 1. During prophase 1, during metaphase 1, during anaphase 1. Those are the big events. Telophase 1, we're going to have the nuclear envelope reform, so we'll have 2 nuclei there, and it'll eventually, after cytokinesis, be 2 daughter cells. And they're going to be 2 haploid daughter cells. Even though it looks like there's 2 copies, remember this is 1 chromosome, and this is 1 chromosome. So there's only 1 of each there. So that's a haploid cell. So there's 2 haploid daughter cells. Now, we look at meiosis 2. So remember, we have to divide twice for meiosis. We have meiosis 1 to form these 2 daughter cells, and then each of these 2 daughter cells are going to divide again in meiosis 2. And this time it looks a lot like mitosis. You have prophase, metaphase, anaphase, telophase happening just like they did in mitosis. So prophase, the nuclear envelope is going to dissolve again, and you have those centrosomes go to opposite sides there, and the spindle forms. This is metaphase 2. So metaphase 2, the chromosomes align. This time they're single file because they're haploid. So those chromosomes are going to align, and then the centromeres will dissolve, and Ana, the mean girl, is going to pull them away. So the sister chromatids will be pulled apart, and the centromeres will dissolve. That's anaphase 2. Telophase 2, we're going to have some new nuclear envelopes form. So nuclear membranes are going to reform. You're going to have the nucleus again. And then after cytokinesis, we're actually going to have 4 daughter cells. And the really cool thing about that is each one of those daughter cells is completely unique. Because of crossing over independent assortment that happened in meiosis 1, each one of those is going to be different. And when you go to study, this is a really great slide. I would use this one and walk yourself through it. So prophase 1, it shows synapses for crossing over. And actually, this independent assortment should have been with metaphase 1. Anaphase 1 is where you have from diploid to haploid. Okay, so in meiosis 1, homologous pairs are separated, reducing the chromosome number by half. In meiosis 2, sister chromatids are separated, producing 4 haploid gametes. So what's separated in meiosis 1? Homologous pairs are separated. What's separated in meiosis 2? Sister chromatids are separated. Make sure you keep those separate. Okay, and there's a whole bunch of slides here to show you in different ways. This one up here, where to go? Yeah, this one right here is pretty interesting because it's going to put meiosis and mitosis side by side so you can see some of the differences. Like in this one, centromeres survive anaphase 1. They don't dissolve in anaphase 1. Remember, centromeres don't dissolve until anaphase 2. Whereas in mitosis, centromeres dissolve in the only anaphase there is. But anyway, you can go through each one of those differences. That'll be something to look for on the test. And then this is a picture version to show you the difference between meiosis and mitosis. So this one shows prophase, metaphase, anaphase, telophase going all the way through. Whereas this diagram over here shows you prophase 1, metaphase 1, anaphase 1, telophase 1. And then it just kind of jumps and it says meiosis 2 down here. But don't forget, it still goes through prophase, metaphase, anaphase, telophase a second time. Okay, so just to recap what I talked about in the last video, where we had, let's see, here's an adult right here. We're going to look at an adult. And the adult is diploid. Both the mom and the dad are going to be diploid cells. However, in certain parts of their body, they're going to undergo meiosis to produce sex cells. So we'll have haploid sex cells produced by meiosis. Sex cells are produced by meiosis. Those two sex cells come together, they're two haploid cells. One haploid come together to make 2N, which is diploid, and that's fertilization. When sex cells come together, that's fertilization, produces a zygote. Now that zygote is going to develop and then grow during the process of mitosis. It still stays 2N this whole time, diploid, 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 but it's getting bigger and growing and developing and even repairing. That's mitosis. Sexual reproduction, meiosis, the coming together of gametes, that's fertilization. Let's see. So sometimes the separation of sex cell or the separation of homologous chromosomes does not go as it's supposed to go. Sometimes something goes wrong during meiosis, and that's called aneuploidy. It's a change in the chromosome number. So let's say right here it's showing you that instead of having a copy from mom and a copy from dad, one in each of these daughter cells, instead they both stayed in one cell. And down here it happened in meiosis 2, and up here it happened in meiosis 1. Anytime something goes wrong, it's going to cause a change in the chromosome number, and that's called aneuploidy. Euploidy is going to be the correct number. So we can actually see when there's a change in the chromosome numbers by using a karyotype, this word right here, karyotype. It's a way of displaying chromosomes arranged by size. Sometimes you'll have a sticky chromosome and there'll be too many. When there's too many, when you have 3, when you're supposed to have 2, there's a trisomy. So this one right here has a trisomy on the sex cells. This one actually has 3 X's. And then sometimes there's a monosomy when there's only one of a particular type of chromosome. Sometimes you'll see this in sex cells as well. Now, if this were to happen on like chromosome 1, 2, 3, that's any of these larger chromosomes, there's a whole lot of genes attached to those chromosomes. So there's a much higher probability that that will not be a viable individual. So they're most likely would not develop at all. However, as you get down to these smaller chromosomes, then you're more likely to see an individual develop from that. Let's see, we've heard of trisomy 21, that's also called Down syndrome, and that results from 3 copies of chromosome 21. Okay, and then you guys can look through these. I only have a few minutes on this video. Okay, so one thing I wanted to go over, so when there's a change in the chromosome structure, what that means is not an entire chromosome coming over, not a trisomy or a monosomy, not 3 versus 1, but a little piece of a chromosome missing. You could have a deletion where just a little piece is missing. One or both ends of a chromosome might break off. That would be a deletion. A duplication is when somehow when they were being copied, the chromosomes were being copied, one section might have been copied twice. So there would be a presence of a chromosomal segment more than once in the same chromosome. Accidentally got copied twice. And then sometimes you'll see something like a translocation, where a segment from one chromosome moves to a non-homologous chromosome, meaning it's not in the right spot. It got put in the wrong spot. These are all things that can happen to a chromosome to change the actual structure of one chromosome. It can happen in things because of things like environmental agents, radiation, chemicals, viruses, and there's some other factors as well. So this is a visual to show you the same thing that we just talked about. A deletion in this one, this section A, just completely broken off. A duplication, you have D and E twice on this chromosome. Inversion, they were put in different places, and translocation, they were put in different places. Both of those bottom ones are types of translocations. Okay. And all of these will cause different changes. And some of them lead to different diseases that we can talk about. But I am going to have to end this video right here. Make sure you review the different steps of meiosis. Oh, here I can show you this real quick. This is a change in the sex chromosome. So this one right here, if you were to see this on a test or something, you could see that this is a monosomy. There's only one chromosome here instead of 2 for the sex chromosome. Over here, this is the sex chromosomes, and there's 1, 2, 3. So that one is a trisomy. This one results in Turner syndrome, and this one results in Klinefelter syndrome.